Hello, this is Dr. Mandar Deshmukh. My lab works on understanding structure and function of proteins involved in RNA pathway, which assists DICER in regulating the gene regulation across organisms, all the way from C. elegans to Arabidopsis thaliana. We solved the structure of RD4 in C. elegans and CRC, which is 32-proline protein in Pseudomonas syringae. We looked at the DRB4 in Arabidopsis thaliana. In addition to that, we looked at the DRB2 and DRB 7.2 complex. We also use a lot of non-NMR techniques like ITC, IMSA. Recently, we also found liquid condensates into the RNA bound system, and we also identified that very similar looking domains have very interesting dynamics that makes them distinct in terms of the conformational space that they sample. The heart of our NMR lies a superconducting magnet operating at 14.1 Tesla and connected with a cryogenically cooled probe connected again to a high-end electronic devices. When the sample is shuttled to the core of the magnetic field aligns the magnetic vectors of the sample, giving rise to the spectrum. And this spectrum is eventually interpreted in multi-dimensions like 2D or 3D NMR spectroscopy. The chemical shifts appearing in 2D NMR are further assigned using NMR methods to lead. Here's an experiment called as HNCA-CB, which is a three-dimensional spectrum which gives the chemical shift correlation of C betas and N and H, and one can clearly span through the planes and assign the spectrum to lead to the chemical shift assignments of the backbone residues. Similarly, one can do the side chain residues and find the distances between the spins. Once you have the distances between the nuclear spins, one can go around and start calculating the structures. The NMR structures are derived at the physiological conditions, which makes them far more reliable in terms of their functional ability, and one can actually map the residues that bind to the ligand or look at the structure as an ensemble where one can find out the flexible parts of the protein at a very fast time scale. At the same time, NMR allows one to sample the conformations that are invisible to other spectroscopic or structural biology methods. Even low populated excited states can be figured out from newly developed experiments called as 15N CPMG R exchange as well as chemical exchange saturation transfer. These NMR observations can be coupled with the molecular dynamic simulations to figure out the real picture of the structural states that are happening in a exchange time regime. And in our lab, we employ all of these methods together to understand the structure and dynamics of macromolecules. We follow the exercise of having a lot of interactive sessions to train students in spin physics and vector algebra so that these concepts of solution NMR spectroscopy are understood by each of them so that they can be applied to understand the chemical nature of proteins and nucleic acids in the study, and subsequently extrapolate the biological role of these systems at the organismal level so that we can contribute to understanding of heterogeneity in the RNA pathway across organisms and its evolution.